Thank you very much. Good afternoon. I'm Neil Fowler. On behalf of all of the employees of Liquidia, we're honored to be here today, and thank you very much for the Needham team for inviting us to present. I'm going to walk you through our usual forward-looking statements and disclaimers, but jump right into an overview of the company for you today. A little bit about the company for those of you that don't know a lot about Liquidia. We're a late-stage biopharm company located in Research Triangle Park, North Carolina. We have a great proprietary technology we call PRINT. You just actually recently heard the prior speaker speak to that. PRINT is a fascinating particle technology. You see an example of our lead product, LIQ861, on the screen here. And with PRINT, what you're able to do is, in a precision fashion, control any aspect of a particle, whether that be size, shape, chemistry. And what that, in essence, allows you to do is very specifically tailor the profile of any therapeutic that you like to deliver into the human body. It has no limitations with regard to route of delivery, with regard to molecule use. We've routinely worked with small molecules, proteins, even nucleic acids. And it's a very fully scaled platform, all located in Research Triangle Park in North Carolina. With that, we started a journey recently to go in our own product development. I'll spend the most time today on our lead asset, which is LIQ861. I'm going to share with you today pivotal data that we're happy to be in late-stage preparation now for sharing with FDA at the end of the year. LIQ861 is the first of its kind dry powder inhaler for the treatment of pulmonary arterial hypertension, which is a very attractive market. We'll talk a little bit more about that. And we have met our phase 3 primary endpoint. So again, we are rapidly preparing our NDA now, and that will be submitted to the FDA by the end of this year. 861, though, is only the beginning. We'll spend a little bit of time at the end on our second clinical stage asset, LIQ865. For those of you that know the Pacira and Heron story well, this is a bupivacaine play where we're able, in this case, to use our PRINT technology to develop a product that will last for 3 to 5 days in terms of post-surgical pain relief. And we will be phase 2 ready with this asset by the end of this year and look forward to our road ahead with 865. And our long-term goal here is ultimately to create a pipeline of molecules and very attractive actives on the backs of PRINT. I'm sure all CEOs will tell you this, but I'm very fortunate to have a terrific team of folks surrounding me. Without going through all of the folks here, I've got a couple of them with me today, but you'll note their experiences down on the bottom. It's a group that's really been there and done that and gives us a lot of hope as we look to our future now as we begin to ponder not only R&D but also commercialization. So this will frame really the presentation. I'll spend the bulk of the time today on our lead asset, which is LIQ861. We have just, as I mentioned, completed a phase 3 study, which is our single pivotal study FDA asked us to do for filing our NDA. And we have worldwide access to this. It's a 505(b)(2), and in essence, we're through with all patient enrollment now. The only thing left for us to report out, which will be a data set this quarter with regard to our pharmacokinetic data. I'll also spend a little bit of time, not as much so only because of being an earlier stage in our pipeline, on LIQ865. We have completed 2 phase 1 studies on this, and as I mentioned, are preparing for our phase 2 work, which will begin in 2020. So jumping into 861, a little bit about the disease first. For those of you that are unaware, this is unfortunately a chronic and progressive disease of which there is no cure to date. It is a disease of the lung primarily, and what happens here is through time, a vasoconstriction develops in the lungs, causing the patient to have a more difficult time breathing. And as the disease continues to progress, the right heart is pumping against this vasoconstriction, and that's ultimately what takes the patient. It's a fairly well understood set of mechanisms that occurs, and it's a host of mediators, and one of those that is central to this theme is called prostacyclin. And we know that in general, if we have a deficiency in prostacyclin, that leads to more of this vasoconstriction. So the name of the game is how do we optimally replace prostacyclin into the body to head off this vasoconstriction and buy quality of life for the patient. It's a very large market here in the U.S. It's just shy of about $4 billion. And as you look at this pie chart, you'll see the prostacyclin portion indicated in dark blue. You'll also notice a lot of innovation. You'll see prostacyclin delivered in a variety of routes, oral, inhaled, as well as parenterally. But the real issue here is we still have not really found the optimal way to deliver prostacyclin. When we think about it being a disease of the lung, in a perfect world, you deliver prostacyclin to the lung where you want it to be, but not cause any of these off-target problems that can occur with so many medications. And to shed some light on that, this gives you a feel for how the disease today is ultimately treated. Typically, patients start in the early stages of the disease on oral medications. These work quite fine in the early stages until the disease progresses to the point that doses have to be escalated to the point that patients begin to get side effects and a lot of off-target toxicities. More often than not, it would be at this point that patients would then be escalated to inhaled therapies, and I'll talk more about this in a moment, but today those inhaled therapies are nebulized, which is a convenience issue for the patient. So typically they'll try to stay on orals as long as possible until they have to go to a nebulized therapy. And ultimately, in the last stages of the disease, the doses have to be so high that patients will move to a parenteral form of medication. So we saw this as a great opportunity to tackle this with our PRINT technology in the inhaled space. So the slide you see in front of you here today is the state of play as we know it in PAH today, and there are two products. The product on the right, Ventavis, was the first product launched years ago. Again, a nebulizer, which is cumbersome in nature. For those of you that are familiar with nebulizers, you'll know that they are multiple pieces of equipment that have to be assembled and disassembled throughout the day. They have to be cleaned and all require some form of a power pack, whether that be battery or electrical power. Importantly, too, here is these patients go through a stigma associated with that, in the essence of having to carry a bulky piece of equipment around with them during the day, which in some ways draws even further attention to the disease. And Ventavis, a big drawback from the beginning was not only the nebulizer itself, but as you see in this first bullet point, the fact that the drug had to be given 6 to 9 times a day. So quite cumbersome in nature for the patient. The second product that was launched by United was a nebulized prostacyclin called Tyvaso. And Tyvaso's big claim to fame, as you see depicted here, was the dosing regimen, and we, in essence, switched from a 6 to 9 times a day dose now to a 4 times a day dose. And in fairly short order, Tyvaso garnered most of the market share. Despite the drawbacks of nebulized therapies, both of these products account for just shy of $500 million in sales here in the U.S. today, with Tyvaso accounting for north of 80% of the market in totality. So we began to look at this in the sense that there had to be a better way. And again, we approached this in the sense that there were a lot of drugs that worked well, but again, this optimal delivery of prostacyclin into the lung could be improved. So this essentially on the slide is kind of the three-pronged stool that we built the foundation of 861 on. First off, we wanted to ask ourselves, what was the optimal active to be using here? And we chose the prostacyclin treprostinil. Treprostinil is the active ingredient that's in Tyvaso. It's well understood. It's been approved previously by the FDA 4 different times for 4 different types of formulations. So the question in our minds here was not one of treprostinil and its efficacy. That was understood. It would be just a question of safety, and that offered us the opportunity to think about a 505(b)(2) approach. Secondly, had to do with the PRINT particles themselves, and you see depicted here in the middle of the slide the actual particles that we use with our PRINT technology. You'll note the exquisite nature that all of these particles are exactly alike. And that derives itself from our technology, which I'll speak to a little bit later in the presentation. But there was a specific design in mind as we thought about these particles. If you think about particle size, this is very, very important in terms of lung delivery. If you operate under the assumption that particles that are too large never make it down into the lung or get caught in the upper airways or don't even make it out of the device, that is not an optimal way to deliver a drug. And on the converse of that, if particles are too small, when a patient exhales, they blow the particles right back out into the air. So it's been fairly well understood that in that 1 to 5 micron range, that's ultimately the size you want to get drug into the lung. And very importantly, if you could hone in on a 1 to 2 micron range for deep lung deposition, that would be an optimal place to play. With PRINT technology, we're able to create particles all of the same size, and that's what you see depicted here, where these particles are just north of 1 micron. Secondly, you'll notice the shape. Actually, this is a very innovative approach we took where we had done some animal imaging work early on to look at lung deposition, and the shape you see in front of you is a mimic of a very common type of pollen that is in the air. And if you think about how pollen disperses uniformly in the air, kind of a relevant topic for this time of the year, you'll note that we saw this also on our animal lung deposition data. So it was very clear to us that this particular size and this particular shape would be optimal in delivering a therapeutic. And in essence, what we do is then take treprostinil and load it into this particle structure, which is mostly sugar, a form of sugar, and acts quite well in relation to the last piece of this stool, which you see on the right, which is the device. This is a device. I've got an example here in my hand for those of you in the room. It's a very simple device, much smaller than the nebulizer that's out there, and very simple for the patient to use. This device is already commercially available in the U.S. and Europe. It's made by a company out of Italy called Plastiape, and used for a variety of beta agonists, etc. This inhaler is approved both in the U.S. and Europe. And in essence, all the patient has to do is take the top off. Our particles are in a capsule. That capsule is deposited into the device. The patient closes the device. They puncture the capsule by pressing these 2 red buttons and inhale. 1 to 2 puffs. They empty the shell back out. They're done. A big improvement over nebulized therapies today. And our belief out of the gate was that this would be a much more convenient and attractive way for patients to have their disease treated from an inhaled standpoint. But the best for us was really kind of yet to come. So we put the product in human testing. You see depicted here our phase 1 results, which were quite compelling. So obviously we wanted to see a nice dose response in terms of blood levels of treprostinil, which you see. You'll note the drug strengths depicted under the x-axis, and it was a classic ascending dose study in healthy volunteers. The original protocol of the study had been designed to go to 100 micrograms, and the reason for that is the maximum tolerated dose for Tyvaso is 84 micrograms, and we figured we had most of that spectrum covered. However, when we got to 100 micrograms, we had still observed no serious adverse events in these healthy volunteers. Our steering committee recommended that we push the dose to 125, which we did. Still no serious adverse events, and ultimately we went to 150, and still no serious adverse events. So we were operating in a belief here that not only did we have a drug that was convenient for patients, but we're clearly showing the ability to dose higher. And if you think about how the disease is treated, this would imply that potentially we had the ability to in essence, begin to grow the inhaled market as we knew it today by potentially prolonging the inhaled portion and postponing the need to go to a parenteral therapy. So FDA allowed us the ability to forego a phase 2 study, and we moved straight to phase 3. They asked us for a single pivotal study that turned into a study we call INSPIRE, and you see the protocol of this study in front of you. It's an open label safety study. Again, this is a 505(b)(2) because we know the active so well. These 100 patients, in essence, come from 2 cohorts. One cohort would be direct Tyvaso switches. So these would be patients that wanted to go from nebulizer to DPI. So we, in essence, switched them directly over to our device. The second cohort of patients were what we call add-ons. So these were patients that were on oral therapies had not yet been on an inhaled prostacyclin and for the first time wanted to do that. So typically earlier stage disease patients that a nebulizer would not typically treat, and they would come on to the drug. Again, a safety endpoint. We were asked to look at a 2-week endpoint, although the primary endpoint of the study in our minds was 2 months, given that this is a long-term disease and we wanted to follow that. But we also built exploratory endpoints that you see here in the study that are more traditional in the sense of PAH endpoints that you'd want to see. So 6-minute walk, quality of life scores, things that physicians would want to ask about, we built into the study as well. We plan to track these patients while we've already hit our 2-month primary endpoint. We plan to track these patients longitudinally. We want to provide these data for the community at large, and we will leave these patients on drug through commercial approval. So it will be a longitudinal long-term study, and you can expect to kind of hear more results of the data from the study as we go through time. And also, very importantly, they also asked a subset of these patients to be put into a PK sub-study, which in essence it takes a look at an equivalent Tyvaso dose as we convert it to an 861 dose. Those patients are now recruited, and we'll be reporting again those data out here in the second quarter. So a couple of slides on the results from the study. We have reported this out most recently last week with some of our exploratory endpoints too for the first time. And again, here's first a cut at where the patients came from as well as how they stayed on drug. And there were some interesting takeaways here that we saw in the study. For those of you that know the disease, as you talk about Class 2s and Class 3s, Class 2 patients tend to be milder and earlier stage. You'll note here that we had a very high percentage of Class 2 add-ons. In fact, we had to turn away quite a few patients in the study at the end, somewhat because of demand issues here. And the nice implication here is that these were patients that had never been on an inhaled medication in PAH and were willing, when given a choice of staying on an oral or going to an inhaled, chose a DPI for the first time. This would have been territory that they would not have picked a nebulizer in the past and chose to go on their first prostacyclin being in an inhaled version. So you'll note there were a high number of folks in this cohort. Very importantly, this led to our early enrollment and it being way ahead of schedule in terms of where we ended up on this. And it was very encouraging to kind of give us validation on our premise that this would be used up front in the disease. The second thing you'll note is in the blue bar, not only did patients come onto drug really in an enthusiastic way, but they stayed on drug. You'll note whether we're talking about add-on patients or whether we're talking about Tyvaso transitions, the dropout rate was very small, and in fact we had patients still on drug in both cohorts together at 93% at 2 months. So very small dropout rate. And one that's been consistent very much so with earlier trials, and one we're very encouraged by. So the takeaway out of this is we're encouraged that LIQ861 may have great utility here as a first-line prostacyclin. It's not just a replacement in our minds for nebulized therapy, but first-line potential for prostacyclin in the treatment of PAH. This also then gives you a cut at the 2-month mark of the safety data, and we're happy to report we met the primary endpoint from the INSPIRE study. We still to date have no serious adverse events reported from the active drug itself. We still to date have not reached an MTD. We have patients north of 150 micrograms and doing quite well. The side effects that did occur that were treatment related occurred early and were tolerated quite well by the patient. And this gives you a feel in this chart. It's a little bit of an eye chart here of total report out of safety, but the far column on the right gives you in composite form all of the adverse events. These are actually quite mild by inhaled prostacyclin standards. And as you break that down, you actually see quite an interesting phenomenon where the middle column here gives you a feel for the transition patients that came over from Tyvaso. You'll note those are much lower side effects where the patient is already used to a prostacyclin and already used to an inhaled therapy, so very well tolerated. what you see is a slightly higher number of side effects, but in a sense that you would expect here with add-ons. These are patients that have never been on a DPI and have never had a prostacyclin. So as you would commonly think, as you go on a DPI for the first time, you're inhaling a powder, you feel that sensation, you have to go through that adjustment phase. Prostacyclins also have their set of adjustment criteria you have to go through on side effects there. These were noted. But what was, in essence, really encouraging about this was the patients, again, got them early. They went through that first couple of weeks of transition and stayed on drug. So this is a durable therapy that we're very encouraged by, and again, through time, we will continue to report out the data from this study. So where do we go from here? We're very encouraged by the opportunity that LIQ861 presents. Today, we're going after a WHO Group 1 indication that you see depicted in this dark blue bar. Our plans at Liquidia are to pursue commercialization on our own here in the U.S. It turns out it's a very efficient launch, and that, in essence, you need about 50 sales representatives to cover the U.S. Outside the U.S., all of the patients are there just like here in the U.S. However, Tyvaso is not approved in Europe, so we will seek a co-development partner and a commercialization partner for our efforts ex-U.S. But very importantly also, long-term, we see expansion. There's a lot of discussion in the PH community, I'll call it, of getting beyond this WHO Group 1 classification and going into larger groups of patients that are more what we would call pulmonary hypertension. So we are supportive in the long run of expansion beyond WHO Group 1, and again, with a long patent life on this product, look forward to being able to take full advantage of that, both geographically as well as in a WHO Group classification. To put a bow around this, our plans again are to report out the PK data this quarter, and then this sets us up nicely for the NDA being filed by the end of this year, and then we'll await FDA as we prepare for our commercialization efforts. So bridging to our second product, LIQ865, I'm going to transition you now from an inhaled medication to an injectable medication, and this having to do with the whole field of post-surgical pain relief. So again, as I'd indicated earlier, I think a lot of people may know the Pacira Heron story, and in short order, that story is built off of an active product called bupivacaine. Bupivacaine has been around for decades. For those of you that aren't aware of bupivacaine and what it does, it would be very typical in the U.S. that you would have a surgical procedure and be injected with bupivacaine as the surgery ends and you're rolled out of the operating room. Think of it as a comparator like when you get Novocaine or something in the dentist and it numbs the area for a period of time. Bupivacaine works quite well. The drawback to bupivacaine is that it only works for about 6 to 8 hours. The patient gets breakthrough pain shortly thereafter. And herein lies a lot of the issues because, as we well know now, about 6%, depending on the database you believe, of these patients will get an opioid and 6 months later still be on an opioid. So a well-intended purpose of pain relief, but unfortunately one that goes awry as we think long-term. So the name of the game has been, as we think about it through time, how do you optimally create a longer-acting form of pain relief? And that's a problem that we kind of set out to do. We looked at Pacira. Pacira took bupivacaine and put it in a liposomal formulation, which improved the performance to roughly a day of duration of effect. Our colleagues at Heron have combined it with meloxicam to create a version that appears through their phase 3 studies to be a 2 to 3 day formulation. But there's still this tremendous unmet need when you think about the need for hips, knees, cracked chest, to have a longer-acting pain relief medication coming out of a surgery. So our belief was, how can we apply the PRINT platform to bupivacaine and see what we can generate from there? So our target product profile here was a 3 to 5 day formulation. We wanted very quick onset. We wanted easy to inject and easy to use in terms of site of care. And importantly, with bupivacaine, you want to avoid what we call dose dumping. It turns out with the canes in general, if you give too much too fast and you spike the Cmax, you can trigger a set of arrhythmias and cardiotoxicities. our formulators were tasked with calming that to make sure we didn't get a burst release. So we went to Europe for our first phase 1 study. This study was performed in Copenhagen, which is a turnkey center over there for speed that we use, but also one that Pacira has used in the past. And great study results that led us to a lot of excitement. There were essentially 3 endpoints we were looking for in this study. The first of which had to do with the ability to increase blood levels of bupivacaine through time. So you'll notice again, healthy volunteers, ascending doses under the x-axis, and indeed as we increase the dose of 865, you see increased levels of bupivacaine through time, which is what we wanted to see. Second takeaway that we were encouraged by, we avoided the dose dumping I referred to a minute ago. You'll notice that there's a quick onset, but it's not a spike, and then the drug tails from there. And the drug, in fact, was very well tolerated as we injected this into healthy volunteers. The third point, while I don't have the number data, we reported this at ASRA, though, earlier this year, has to do with the last bullet point on the side here. Not only did we want to check the pharmacokinetics, but we also looked at pharmacodynamics. So we wanted the patient to report their version of pain relief that they felt. There are different ways you can stimulate pain via mechanical and thermal approaches, and we measured this through time. And the results for this were quite encouraging in that there was a clear dose-response effect that told us not only did we get higher blood levels through time, but we had pain relief out to 3 to 5 days. This told us that we had reason to believe on the product we had in front of us. So we did a confirmatory study back here in the U.S., which was completed last year. And now as we look to the future, we plan to have a phase 2 ready asset with 865 as we move into 2020. We have some requisite tox work that we've got to complete this year, and that will essentially look at a fairly well laid out regulatory path as we look at Pacira and Heron that would suggest a soft tissue and a hard tissue surgical model as we get into phase 2. In closing, just a couple of slides about the technology itself. We get a lot of questions about PRINT, how it operates, how do you do this, and a few things around how scalable is it. I'll send everybody to our website if you want to see an actual animation video of this. It's a much better way to show this than in still imaging, as I'll show here. But in essence, this technology comes out of the semiconductor industry. And what we do is as I think many of you know, if you take a silicon wafer, while it looks smooth on the surface to the naked eye, you'll know that there are a lot of surface features across that. And in essence, what we do is harvest those features into particles. So we begin as depicted here for cartoon's sake. As you see here, this template that's gray has cylindrical posts. So let's say we want to make a cylindrical particle. Those posts are etched across the silicon wafer. Our special sauce is the green blob that you see there is like a liquid fluoropolymer, super Teflon if you want to think of it that way. We pour that across the silicon wafer, we harden that, and we peel it back. And when you peel it back, and if you think about it now, you have an inverse mold of those cylindrical posts. It's like an ice cube tray or a muffin pan, if you want to call it that. And now we have the ability to fill each of those cavities with an active or active plus excipient, as depicted in red. And as we harvest that out, for the first time ever now, you're left with a two-dimensional array of particles where every particle is exactly like the one beside it. And this gets past things like liposomal approaches where everything's spherical in various sizes. It gets past things like spray drying, milling techniques where they're in essence somewhat crushed particles that have a variability in terms of size and shape. And as you think about loading a therapeutic into those particles, you take a bell curve of particles and a bell curve of outcomes and in essence begin to think through a straight line. And I think you've seen that now clinically with 861 and 865, a couple of examples of how we can do that. You see depicted down below just 3 really simple examples of some of the particles we can make. We can make particles as was indicated in the prior presentation. We've done particles in the eye up to several millimeters in size. We've done particles down into the 50 nanometer range. We're only limited on the downside by how far the semiconductor industry can go. So we can do a wide array of sizes, shapes, anything you can imagine, and we take cues from biology to lead us to what those particles should look like. The greatest part about the technology in our minds, too, you see in front of you, is the scalability of this. So we have adapted this to a roll-to-roll capability, so very understood technology now in terms of scaling, and it's essentially wider webs, faster throughputs, and more machines in terms of scaling. We have the 3 machines you see depicted in front of you at our site at Research Triangle Park in North Carolina. Moving from left to right, the line on the left is a heavy R&D machine that can be interchanged for a variety of actives. The machine on the right is being validated now. That's a pure commercial machine and quite simplistic compared to the R&D machine. And then the machine in the middle is a hybrid. So we are scaled for production for both of our first 2 products now as we move forward, and it's a very straightforward, low COGS, low CapEx production capability that we have with this technology. So in conclusion, we've got an exciting period of time underway right now. It's about execution for Liquidia. It's been an active year for us. We reported the 2-week safety data after J.P. Morgan that FDA had asked us for with regard to 861. We recently, as shared last week, reported that we met our primary endpoint of 2 months for the INSPIRE trial that we shared, along with, for the first time, some of those exploratory endpoints that I mentioned earlier. We have initiated now our tox studies for 865 to prepare us to be phase 2 ready by the end of the year. And we'll report out the PK results this quarter as it pertains to 861, all in building toward an NDA filing by the end of the year for our lead product. So thank you for your time. I will pause here. I know we've got a minute or 2 if anybody has any questions, and we appreciate your interest today. Thank you. Thanks very much. You over here?